Good evening, everyone. We will start this session. It's my pleasure to welcome you all to this special event organized by Scioverleaf on the occasion of International DNA Day. We are gathered here today to celebrate the remarkable discovery that has made the revolution in the fields of science, medicine, and even our understanding of ourselves. That is, the discovery of the structure of DNA. International DNA Day is an annual observance celebrated on April 25th to honor the discovery of the double helix structure of DNA in 1953 and the completion of Human Genome Project in 2003. This day serves as a tribute to the pioneering scientists James Watson, Francis Crick, Wilkins, Rosalind Franklin, Friedrich Miescher, whose groundworking work laid the foundation for modern genetics and molecular biology. So the discovery of the DNA structure in 1953 transformed our understanding of heredity and genetics, revealing how genetic information is stored and passed down. This breakthrough impacts fields like medicine, agriculture, and forensic science. The completion of the Human Genome Project in 2003 further advanced genetics by sequencing the entire human genome. This provided insights into humans' DNA role in health, paving the way for personalized medicine and genetic testing. DNA Day is not only a day of celebration, but also an opportunity to raise awareness about the importance of genetics and genomics. It encourages dialogue and the education about DNA, genes, genetic disorders, and the ethical, legal, and social implications of genetic research and technologies. I'm proud to introduce our team members, Savitha Madhusudanan, the founder of Scioverleaf, Tamilarasan, another founder of Scioverleaf, myself Satya, Chief Coordinator, and Tarangini, other Chief Coordinator, and we have content writers Sajia, Vishwan, and Vaishnavi, who are the backbone of our team. I hand over the session to Tamilarasan to invite our guest. Thank you.
Thank you, Satya. Greetings to all the attendees and esteemed guests. With myself, Tamilarasan, founder of Scioverleaf, and it is an honor to introduce a trailblazing professional whose profound impact on the field of genetic engineering underscores the essence of scientific excellence. Today, we have the privilege to welcome Jeevitha C M in our midst, a scientist, innovator, and a visionary leader whose contribution in the field of genetic engineering are truly remarkable. Jeevitha's passion for science was ignited at a young age, propelling her on a path of relentless pursuit of knowledge. Graduating from SRM University with both her BTech and MTech degrees in genetic engineering, she demonstrates an unwavering commitment to academic excellence in scientific exploration. Throughout her career, Jeevitha has exemplified dedication and integrity in every endeavors. As a founder of Biovagon, she has pioneered groundbreaking research and development initiatives utilizing her expertise in computer-aided design and a plethora of other skills to drive innovation in her field. Her visionary leadership has propelled Biovagon to the forefront of scientific discovery, positioning the organization as a beacon of excellence. Additionally, Jeevitha's tenure as a research scientist at University further underscores a commitment to advancing scientific knowledge. In her role, she brought her unparalleled expertise to the profound, contributing transformative research endeavors that has the potential to share the future of genetic engineering. As Jeevitha embarks on her Ph.D. journey, her passion for science, her dedication to excellence, and her visionary leadership serves as a beacon of inspiration to us all. Let me extend a warm welcome to Jeevitha C M, celebrate her remarkable achievements, and join hands in embracing the limited possibilities of scientific exploration. And I'm personally very thankful for her short invite and her acceptance of a short invite. Jeevitha ma'am, the dice is all yours, ma'am.
Thank you for the very nice introduction. You have told so much about me. And first of all, I want to congratulate you guys because you're in bachelor's and at the very beginning, you started something different something different, something additional apart from your career. Like this is going to be like my presentation is all going to be very common, very basic, and like the way I wanted to give advice when I was in BTech. So I'm going to share uh, like it's 5 years difference between you guys and me, and what are all the informations I could I wish to get when I was in BTech. So I wanted to share like very basic career insights to you guys, so that when you're choosing something in further, when you're completing your bachelor's or when you're completing your master's, so you need not to struggle the way we are struggling now. So that is the main motto. And firstly, like you have started to blogging everything from very beginning of your bachelor career, and very congratulations. like Not only so first of all, uh, we are genetic engineers or biotechnologists, and somehow we have come to this career with either fighting with family that we have to join biotech or genetic engineering, and in some cases we will be trying for NEET, and the second option will be biotechnology for us. But most of the people who choose biotech or genetic engineering is like they want to be in research, they want to like with a love only we came for genetic engineering, because I fighted with my family to join this course. Like 6 years back, nobody knows what genetic engineering is. Now after COVID, like everyone speaks RT-PCR, everything. But before 6 years, nobody knows what is genetic engineering, what is biotech, what is the work we will do. So all the career insights we are going to discuss. And first of all, congratulations to all your team members, every one of you. And in further steps also, I'll be very happy to help you in any ways, like you need my support or guidance, whatever the way you guys are expecting. I will be always available for you to help you. And so I'll share the presentation I have made specially for you guys, so we can start the session.
Sure, ma'am. Thank you so much for coming to the event, ma'am, in a short notice.
Sure. So can you guys see the presentation?
Yes, ma'am. It's visible.
Okay. So this session is entirely going to be you have to answer me what is the questions I'm going to ask. So it will be like interactive sessions. So I need one minute. So what is life without DNA? So I'm not going very basic like what is DNA, everything, but what is life when there is no DNA? So you can it can be interactive session, so you can type your answers in the chat box. So I'll be looking at it what you guys are answering me. Okay. Okay. So what is life without DNA? So when there is no DNA, there is no life, first of all, because whatever we are, whatever the face we are having, whatever the characters we are, so everything is based on DNA. So without DNA, there is no life. Like that's the last point I have mentioned here. And there is a cell confusion, because uh, your cell will get confused where to keep your eyes, where to keep your nose, everything, because there is no proper coding happened in your DNA, and missing family traits, and there will be no inheritance happening, and stuck in the past. Like there will be no evolution of new species or new organisms, and obviously there is no life. So DNA is very important. And like the previously, she has given the nice intro of what is this DNA day and why it is celebrated, so all those things. And today is a National DNA Day. So all we have to do is we have to remember why this day is celebrated, because of Human Genome Project completion and the discovery of DNA. And also we have to celebrate like where we have reached now. Like from the beginning to now, there is a great advancement in the field of genetics and genomics. And genomics is currently leading in all the field. Like you need genomics techniques or whatever it is, the end product will be genomics. And to teach, like we have whatever the research we do, like when we start entering the field biotechnology, everyone's aim will be like we have to discover drug for some cancer, we have to discover drug for something, some particular diseases, but it's like ultimate aim. But what we are doing is research. We have to understand that every simple basic thing we are interrogating, we are identifying is very crucial and very important, because we don't know what a single gene can do, or what is the like simple transcription factors, promoters. So there is nothing very basic, or there is nothing very important in science or in the field of research, because you should not consider that when you are into the field, like everyone wishes like we have to discover the drug for cancer, but that's not that's the aim. But we can't do it like by just doing PCR, genomics, all those things. We can't directly jump into that we have discovered something. What it is there is nothing basic in it. That is the main point I'm telling. Whatever research you are going to do is very crucial. There is nothing like a basic research, simple research, advanced research. Research is itself a very big advancement. So don't take any simple things or any simple experiments you are doing in the lab or whatever it is as a very basic, because everything whatever you do, a simple mistake is also a very important thing, because your mistakes may lead to some other new invention, some other new, you can identify some new function of that compound, you can characterize it in different way. So like keep in mind that whatever you are doing, whatever you are getting errors, everything is very important when you are in lab. So that is the main thing I have to tell. And we have to discuss, we have to tell the common people what is like now when we speak about biotech or genetic, there are many people who can understand what we are doing, and we have to inspire like now it is very advanced due to corona, because whatever COVID has given so much of distractions in life, but when it comes to the field of biotechnology, COVID has given like life to the biotechnology field of people who are doing research, so that uh, the other people will respect us like, okay, she's in biotechnology field. At least they know what we are doing now. So it's a very big change, and we don't know whether this thing will be there for so much years, because that's how our society works. When COVID is in peak, everyone was doing RT-PCR, everyone was doing genome sequencing of COVID, everything. But we don't know how much time this craze will sustain. And I'll also come to the statistical part of how this research thing works. Okay, so my main thing was to help you in the career guidance. And the four things, like I wanted you to start very early, because most of the people I got is like they are working, they are in their second year or third year of biotechnology. So I think this will be very useful to you guys. And why I have taken this career insight is like we don't, when I was doing BTech or MTech, there was no one to help. You have to choose this, you have to choose that. No, there was many complication in choosing whatever you are taking. There is no clear example, because like you completed your BTech and you don't know where to go, whether masters or you have to go for some job. And there is no use of doing BTech in genetic engineering and doing job in IT company. I don't go with that ideology, because maybe some people have taken this biotechnology course, like they need some degree for other aspects of life. But those who have taken this with so much of love, interest, and passion, we need to know what is the next step. Like, because we know in India there is not much opportunities in this field, but I wanted to tell you guys how we can make benefit, or we can make a step ahead in this competitive field, so that
Ma'am,
yeah,
sorry for interrupting you, ma'am. The session is really great. But I think you're presenting, but we're not able to see any slides, ma'am. We're at the first slide.
we can't. Okay. So it's in career development only. I haven't gone to the next slide.
No, actually, until now we had the first slide only, ma'am.
Oh,
We
sorry.
hadn't. Yeah, yeah. So I think you were presenting, but that particular thing was not showing.
Okay, so I'll try again. Now, is it changing?
No, ma'am, it is not changing. It is at second slide.
Okay, shall I continue in this way?
Yes, ma'am. Yes, ma'am. Sure.
So I was speaking in this slide. So our this session is all going to be like I'll be speaking with Jeevitha 5 years back. I'll be giving advice to her what she has to be done. So I think it will give you guys idea, like because we don't know when you guys complete after 5 years there will be more advancement, but you guys have to be prepared in all the ways. Like there is no we can't focus only in one technique or one particular topic in the genetic engineering, because there are many competitive fields going on. So I'll be talking on that. So first of all, I have to ask about like what kind of audience we have, either BTech, BSc, MTech, MSc, or the people who are doing PhD research. So you can comment in the chat box, so I'll see and I'll reply. And can you guys guess what I have pictured for BTech and for MTech and for PhD? And any of you, can you guess why I have given this images? So one has replied as PhD aspirant, and most of you are like from the previous introduction, you were in BTech. Okay. So when it comes to the field of biotech or biotechnology, your life begins, or you're like a complete graduate or someone who know completely, or someone like you are starting your career when you are doing actual PhD or research. So what I have given for BTech is zygote, and for MTech is a fetus, and for PhD is the newborn baby. Because even in the PhD, you will be like learning many new things, and when you have to do something, uh, you have to go for academic field, or you have to get a good salary package in the life science lab, whatever it is. When you have to have a secured life, like after pursuing these courses in biotech or genetic engineering, I consider PhD is very basic thing, because when you complete BTech and when you go out and when you seek jobs, and most of the MTech graduates or PhD scholars who have tried can know that you'll get the work in medical coding and your salary will be 12,000. So you will decide, okay, in MTech I'll get stipend of 12,000 so that I can study, I'll get degree, so it will be beneficial. So you'll do MTech. Even after MTech, when you go to the job market or whatever it is, so the job they'll give you will be very basic. Because why this condition is happening in India is, there are many job markets, there are many growing startups and life science laboratories, but uh, everything in India is like they do most of business related of work. Like there will be no research, because when you want to do research, when you want to join lab, when you want to do research, when you want to publish paper, it's very hard to find such jobs in India. Like why I'm particularly telling in India is because in abroad there are many funded labs, funded companies where you can research, which is very collaborative with the university, so that you will get a opportunity to work in the lab. Because at the end, We want to do research. We don't want to sit in the sales manager or a business analyst or such kind of job after doing all these MTech courses. So that's why like I have given PhD as a newborn baby here. And I want, like before COVID or whatever it is, I want you guys to tell like how you will explain to your relatives or whoever, like when they ask you what you are doing or what you are studying, what is the work you will be doing. So how you will explain those things to your relatives that this is the work I am doing without take 1 minute, without taking reference of, without taking reference of this movie. So how will you tell that you are doing this work? So in most of the people from Tamil Nadu, when you have to explain your relatives or some other people what work you are doing, obviously you have to tell that I am doing work which is related to this movie, Seventh Sense, or which is Ela Marivil. And because when I was starting 6 years back, everyone used to ask me, what is the work you will do? Even when I start explaining that I'll read related to genes, DNA, everything, they wouldn't understand what I'm telling. So everything will, I wanted to tell this movie, like, have you seen this movie? So I'll be doing some kind of work like this. Then, okay, they'll tell, oh, okay. Because this was what they had before COVID. Like, you have to explain what you are studying based on some movie. And in the BTech itself, you have to decide what is your long-term goal. Like after completing your BTech, like you are in almost second year, third year. So what is your next step? You guys want to be in research, or you'll be going for some job opportunities, or you're going to study abroad. So like you have to choose what you guys need. Because I can like there is many pros and cons. It all depends on like your ideology, what you have to go. And when we started to explain all this, like about from this slide, it takes almost 2 hours, because when you have to choose research, there is a separate path you have to follow. When you have to go for job opportunities, you have to enhance some additional techniques in you, or is this beneficial, or you will get a job related to your field. There is no guarantee. And if you want to study abroad, there is also some consequences, like you have to spend so much of money, like you have to take education loan. And even after studying abroad, there is no guarantee that you will get job. There are many consequences on choosing these long term goals, but you should be knowing what is your next step, so that in this journey of BTech you can fix that, you can enhance your skills for choosing your next step. So that is what main thing. So what is the suggestions I like to give is, when you are in BTech, attend all the workshops. Like you may be interested in plant, but attend the workshop which has animal modeling, cancer study, whatever it is. You may be like most of the biotech people, they won't like coding. And there is a misconception that bioinformatics is not completely coding, because many people think that when they say like you have to learn bioinformatics, that doesn't mean that you will be sitting like an IT guy and you will be coding. No, bioinformatics are very basic now. Like without whatever your work, whatever we are going for job or you are going for PhD, bioinformatics is the basic skills they are looking forward. So never ignore bioinformatics, because it is very, very, very important. And you also know that we are not in the era of bioinformatics. We are advanced. We are in machine learning, artificial intelligence. Like we are in advanced development of bioinformatics, like computational. So bioinformatics is very basic. Please never ignore it. Bioinformatics is like you have to take some data, you have to put it in the softwares. Like you no need to do, you just have to use your brain to analyze the result. There is no other work you are going to do. You will get the data from some website database. You'll upload the data in some other database, so you get the output. the way you are analyzing the data, the way your brain is working on the data is the main thing. So there is not so much coding in the bioinformatics. So please don't ignore bioinformatics. From BTech, start focusing on bioinformatics. So that is why I have told, like, attend workshops in all the field. Like, because we don't know when, in which field we get very interested in. Don't narrow your field. Like when you are BTech, explore everything. Go for internships or go for workshop with bioinformatics or animal cell model, cancer cells, whatever opportunities you get, go and utilize it. So when you are in the stage of choosing that, okay, I'm interested in doing gene editing, I'm interested to work in Alzheimer's, so I'll go for neurobiology. So you can have options. Like when you know the basics of all the field, you will have better ideas and better options when you are in the edge to choose something for your next step. And join lab during your free time. So why I tell this is, uh, to make good connection with your professors, basically. Like, uh, you need good connection with your professors, because after BTech or MTech, wherever you are going, you need LOR. If you're going for job, there is no need of LOR. But when you want to stay in academics, or when you need some help in further some or other way, you need to have good connection, or you need to be disciplined in your lab or in your college. So when you get time, go yourself, go to the professor. Like if you want to learn something on animal cell culture, go to the professor who is handling that lab, or go speak with her, tell her that you wanted to join a lab to learn the basic things going on in her work, like in her lab. So like, as your BTech student, they'll not give you so much work, but there is, like, she will be back of you. Like, you will be under some professor who will be supporting you always. So that will give you the good impact, and you will have exposure to various techniques, and it will be very beneficial to you guys. And if your aim is to join some jobs, learn coding. Because when you learn coding, I'm not telling about medical coding, I'm telling like basic Python, or learn something which is related to biology. Like, it is very easy to learn coding. Start learning that. Use Linux, Ubuntu, everything. And there is a something called Anaconda package, which is, in that you will found something called bioconductor, which is particularly for biology. So you will have access to everything. The same blast you are doing in the website, you can do that with using coding also. So learn all this basic coding. So when you know some coding languages like Python, Perl, all in the field of biology, you will get better options. Like, you can be a data analyst, or um, something related. You'll get a better job than medical coding. So that is why I'm telling. So all these things are, like, after BTech, to help you guys to choose the better path. So these are the suggestions I could give. Make good connections with your professor. It is very important that like, speak with them. You ask them that, can I come and learn this technique? And don't be in only, if you are from SRM, don't be in SRM only. Go to outer colleges, go attend workshops there, speak with the professors, attend conference. So when you attend conference, you will get opportunity to speak with the professors from abroad. So just attend their lecture. You will just get one doubt. Go and ask them, like, ma'am, why you did this? Why you did that? So they will be remembered you. They, like, some person has asked me this doubt. So you'll have good impact and good connection for further uh, enhancing your career. It will be very much useful. And when you are doing project, so this is very important. As I said, no research is basic. Like, don't ignore it is basic research. I don't want to do. I only want to do something which is to find drug to cancer. Don't choose project in that way. So because when you are in BTech, first do it. I'll say, like, choose an effective field. So you have attended all the conference, workshops, everything now. And you have come to the conclusion that I'm interested in neurobiology. So I'll choose this field. So if you are choosing neurobiology, you have to choose what are all the advanced techniques at presently the papers are published in neurobiology. Because when you are going for abroad, or actually when you are choosing neurobiology as a BTech project, you will be thinking that I can do masters in neurobiology also. So based on that, you have to decide what is the field of study you are doing. So when you are choosing your major project, don't choose like, okay, this professor is having vacancy, I'll go and join there. Like, don't do this minor mistakes. Like, choose effectively, like which you love, which you wanted to research. So choose the effective field of study you want to do. And when you are in BTech, you will have an opportunity to do it in collaborative labs. Don't miss it, because when you do in MTech, you will not get that opportunities from most of the universities. Like, most of the universities will not allow you to go do your MTech project in some other university or a company for 1 year. But when you are doing your 6 months BTech project, you will have an opportunity to work in labs from anywhere, any part of India or some other university, collaborative labs, all those opportunities. Please use that opportunities, explore yourself, go to the new places. Like, to advance your career, just make connections. Like, learn from everyone, enhance your skills. That is what I'm coming to tell overall. So be extrovert. Like, don't think that how can I ask the professor for the project. No, when we start exploring, when we speak what is in our mind, when what is in our heart, then only we can lead that way. Or else we will stop, we'll stuck in the place where we are when we are not expressing our thoughts. So please don't be introvert in this case, because the people who really love research or who really is passionate about biotechnology, please express yourself. So simply you can post in LinkedIn when you have studied one research paper, you are very excited about it and you felt it very very nice. Like, you felt it, wow, this technique is very nice. Express your thoughts in LinkedIn. So make your LinkedIn profile also very professional. Keep updating your thoughts, whatever it is. And go to SAP if your aim is master's abroad. So why I'm telling this is, it will make your path easy when your ultimate aim is choosing abroad. Because when you have to do a master's or PhD in abroad, this is the easiest way you get into European countries or America or wherever you have to go. SAP is the easiest way. But the thing is, you have to spend some amount in SAP. But I guess that it will be beneficial, because when you go for BTech SAP, you can ultimately decide your master's lab, everything. So you can easily go for masters. And when you join masters, you can join in the same lab as a PhD scholar, or you can explore other options in the university, or you can do some collaborative work with your professors. So like, SAP will reduce the time, because when you are not doing, like, for the people who didn't went for SAP, or all their bachelor's, masters, everything is in India. So it ultimately takes almost 1.5 to 2 years to get PhD in abroad, because it is not that easy as we are thinking. And sometimes it is easy when you are all like, you have attended workshops, conference in all the field of study. When you have narrow uh, during the BTech or MTech, you have focused only in the plant domain, and for PhD, if you are focusing something other, it will be very hard. So that is why I'm telling that don't narrow your thoughts or field to work. Explore everything, some or other way. Everything will be useful for you guys, and focus on the domain and skills used in the project. As I said, when you're focusing on neurobiology, uh, like, don't go for the older techniques. Like, in neurobiology, there will be some advanced fields. For example, we can take genomics. So whole genome sequencing, exome sequencing, everything is a very older technique when compared to the present situation, because at present there are new uh, new sequencing techniques, effective, cost effective, time effective, and very precisely sequencing methodologies. So in every field of study, there is like advancement. So look in the techniques which is advanced, so that when after 5 years, the technique you are learning now as advanced technique will be very basic. So it will, like, if you are learning older techniques, we can't be in the competitive world. So when you are choosing some domain, see, like, try to learn the advanced techniques and enhance your skill in that. So I like to tell what are all the skills you can be involved. So next generation sequencing, NGS is very common in all the applications you see, either in LinkedIn, either for PhD, either for job, whatever it is. Sequencing NGS is very common. So try to learn NGS, attend workshops, conference, internships on NGS techniques, and all these other parts I have written is don't ignore anything. When you get opportunity to learn some animal cell culture in other labs, or other universities providing workshop on animal cell culture, mice handling, attend it. Like, put some money in this. Like, for workshops and internships, it will take around 2000, 3000. So I, like, it will be helpful for you guys when you make your resume and you know the skills and you have proof that you have attended these workshops. So it will be very helpful to you guys, because once you guys get passed out from BTech or MTech, you will not have any opportunities to learn. You'll not, there is no such thing like internships or some training programs will be provided for the BTech and MTech. You will be either going to job or you will be going to academic purpose. And when you want to learn also, they'll not give you that opportunities, because they want some candidate who can work for them. Because what will they get by teaching you something? When you are master students, when you are BTech student, when you are registered in the university, so you will be doing 6 months project attending. But what they will get after you completed your masters and you want to learn something, what is the benefit they get from it? So they'll ask you to pay. They'll ask double the amount. Like, they'll ask 50,000 for learning NGS. But after masters, we will be not in that hand to pay so much of money and learn something. So that is the reason I'm telling, like, get the opportunities when you are already in the academic career, like when you are already registered in the academic. So when you are in masters, you have 2 years. So use the opportunities, learn as much as possible when you are in the department, within the department. Same for BTech students, learn animal handling and important microscopic techniques. Like, plant, I don't think like much experience is needed in plant, because all the basic molecular biology things you should be focused, and greenhouse management, all those things. So you can handle that. And bacterial work or coding. Please learn bioinformatics. Don't ignore bioinformatics in any part of your life. And we are in the field of machine learning and AI, that is, which is advanced bioinformatics. So, like, as of now, machine learning is advanced. For and the BTech students who are in second year, third year, everything. So once after 5 years, machine learning and deep learning will be very basic. You will have some other advanced techniques than this. So we can't be in the competitive world. So try learning this first. You focus on bioinformatics, learn the basic bioinformatics, then start learning coding, which means biocoding, and get the knowledge, like basic, what is machine learning, how you can integrate machine learning in the field of biology. So you can do in finding molecular evolution, protein analysis. There is like a precision medicine you can do, and you can identify biomarkers. So, like, even when it is very basic, there are many advantages of using machine learning and deep learning. But don't go only focused on bioinformatics. You need both wet and dry lab techniques equally skilled. Because when you go bioinformatics, whatever data we are getting from bioinformatics is validated using wet lab. So we need both equally. Give importance to both this field, like dry lab and wet lab techniques, equal importance, and learn as much as possible. And the competitive exams in a field, try to attend it when you have to be in India, when you have to be in academic field, when you have to become a researcher or lecturer in India. Try attending this competitive exams, GATE, GRE, NET, and there is something called BIT, biotechnology eligibility test, where you can have some vacancies, fill the vacancies in Department of Biotechnology, like DBT platforms. And you also have a NEST, which is for this is like a screening test. So try attending these competitive exams, like that. It's not to be compulsory. You can attend it any time of your life, but keep that, like have some gate score, some net score, so that it will be it will show your eagerness, or it will show your cracking this competitive exam is also some skill. Like not everybody can do that, so it will be an additional skill in your CV. So try attending this. And I want to tell that how the research field has changed before COVID and after COVID. So we can see that after COVID, there is a increase in everything. So in the first, we can see there is an increase in the investment and funding around 40 billion INR, and the market is also doubled. And even after 5 years, so India will have so much of biotech companies. Like we are considering India as an IT hub, because whatever the company, wherever it is in Switzerland, America, Australia, wherever it is, the most of the IT companies who are working for it are in India. So after 5 years, biotech also will be in same situation, like other European laboratories or European labs will invest so much in India, because we know we have so much of manpower, and they will be doing double the work they do. So there will be very much develop or advancement in the field of biotechnology. And approval time is also reduced by 6 months. And like where we are lagging is, I can tell in this part. So before COVID, the research on COVID was going 5%, but after COVID, it is 65%. Okay, this is good. But the research before COVID in healthcare, like normal other diseases or normal other department of health, is 45%. Now it is reduced to 20%. So that means, like when it is like uh, COVID came and everyone started researching on COVID, and the other research is going down, like 25% is already we are lagging. So what about the other researchers? What about the other important aspects we have to see? There are many other diseases, many other things where we have to focus. But when something is in the market, we started to focus only on that field, but that should not be the aim. Like everything should be given equal importance. And you can see the agriculture, it was 25% before COVID, and now it is only 10% of work is going. And same in the industrial aspects, it is 30 and 5%. So before and after COVID, most of the research happening is on COVID. So but it is not going to stay the same forever, because just now still we are coming out of that COVID. It's almost 5 to 6 years. So it will not stay same. Like not every research will be on COVID-19. We have to again look back, and we have to work on cardiovascular diseases, cancer, malaria, all the basic research we have to start doing. So that is what statistical says. So why I'm telling this is, when something is in the market, people start looking at it, and everyone start doing research at it. So uh, don't don't be in that circle. And when you are doing a BTech project also, there is nothing called basic research. I'm telling this repeatedly, because everyone we who choose biotech, is everyone's aim is to discover some drug. That will be the ultimate aim. Like as a scientist, I have to discover some drug for cancer. Yeah, you are right, but don't generalize or don't make any researchers this is basic. Why I have to work on transcription factors? Why should I work on promoters and enhancers? No, nothing like that. You have to work on every part of the research, because some or other way it will lead to greater beneficial. So for example, the lady who got Nobel Prize this year for mRNA therapeutics, Kathleen, she was working on the very basic research. She was chemically synthesizing nucleotides when she started her career, and nobody supported her. Like they were doing that, it was something related to Department of Chemistry, but she was trying it with her colleagues, like I want to chemically synthesize the nucleotides. And then ultimately she got a colleague who is working on immunotherapy. And that's when she started to learn what is mRNA, what is tRNA, what is the modifications she can do in that nucleotide. And that is what that invention and that progress of her is what led to the discovery of mRNA vaccine during the COVID. So if she has thought that, why should I work in nucleotide? It's a very basic thing. Why should I modify the nucleotide? Like if she could have thought that, we couldn't have that mRNA vaccine when it is most needed during the COVID times. So I tell you guys that don't think any research as basic. Whatever you are doing, do with full passion, full knowledge, like try to gather utmost knowledge of that simple thing, learn everything, and put your all efforts when you really love research and when you really want to do. So that is all I wanted to tell today. And I want to uh, thank Scioverleaf for this opportunities, like to speak with many of the people who love biotech or attending this. So I'm very happy for invited to this session and giving you this talk. And if you guys have anything to ask me, you can ask me.
I see a raised hand. Please go ahead with the question.
So you can ask whatever doubt you have in the chat section. You are in mute, I think.
Ma'am, you are on mute. Kindly unmute and ask your question, ma'am.
Excuse me, ma'am.
Yeah.
Uh, Thank you for your valuable information, and we need updates regarding the immunology as well as the pathology related fields too.
Okay. Like updates in case, like what you guys are expecting.
Okay. Specialization in the particular topics, the immunology as well as pathology.
Like if for masters or after masters.
Postdoctorate, ma'am. Postdoctorate as well as fellowship.
Okay. So I'll look
Mm,
into it and I'll let you know. I'll give my contact information in the chat box so you can connect
Thank
with
you.
me.
Sure,
How
ma'am. Sure. Thanks.
Thank you. So how to select career after MTech genetic engineering? So uh, you have to decide what you have to do, whether you have to go for a job or you have to pursue your PhD. When you have to go for a job, obviously you will get selected to medical coding, but don't use like that's not that's that is not why you started coming to the next step of BTech genetic engineering. So try applying, and uh, based on your project from the master's genetic engineering, try applying in the jobs which is very similar, because you'll easily get. And when you are currently in doing MTech genetic engineering, if you are doing that, try contacting your other professors, learn some techniques from them. Like if you are doing in plant, learn some techniques from the cancer lab, or make good connections or get LOR from the cancer lab. or whatever it is. So enhance your skill in which way you have to go further. So if you want to apply PhD, um, like look into the field where which is exciting to you. So choosing the career is based on yours. You would think taking MSc in bioinformatics is a good choice. Like it's a very good choice actually, but yeah, like if you are doing MSc in bioinformatics, particularly specialized, you will have a step ahead in your race. Like when it comes to job, also when it comes to um, PhD also. But when it comes to job, you will be given preference when compared to other participants. And when it comes to PhD, you have to continue your PhD in something like computational biology, such cases, because they'll also be looking for a wet lab works if it is a lab with a collaborative things, doing both wet lab and dry lab. So when you are doing MSc in bioinformatics, in between, if you think of doing some other lab or some other field, try learning some other wet lab techniques also. So that will be very useful. So when you are doing master's project, include something like half wet lab, half dry lab. So it will give you so much opportunities. As a first year biotech student, what should I do to improve myself and to get into the research field after my UG? So, as I said, you have to join the labs, you have to attend workshops in almost all the field, because you can't decide which field is better for you. Because when you know only cancer biology, what you think is okay, cancer biology is good, because you will not have other opinions about other field and you don't know what you really like. So give yourself that time and give yourself that opportunity to learn all the field, to know basic about all the field, plant biotechnology, cancer biology, and some other, like whatever domains, whatever workshops, internships, conference available, attend everything. So when you know, when you are in third year, when you have to choose some lab, you will feel like, okay, I attended that workshop of neurobiology, or I attended that workshop of something related to bacterial antibiotic resistant. I am interested in this field. So you will have clarity which actually you are interested, so that it will be easy for you to choose your research. And based on that, you can choose what is the skills or what is next you can learn in your masters. Yeah, like medical biotechnology is also good field. Like every choice depends on you, what you guys wanted to do. It's not like this is good field or this is bad. It's like you have to do something which you are actually into it, which you actually love. So you'll be like satisfied when you are doing work. That is what main thing matters. Like for the previously one who asked bioinformatics is good, it will be good for her if she is doing bioinformatics. And if you feel medical biotechnology is good, you will be happy learning it. So you will put so much of focus, so much of time into it. So it is the good field. Like, yeah, entrepreneurship is also good. And like you have to come with the innovative ideas, according to me. So that is why we started Biovagon. There is much more to come from Biovagon, but we'll discuss that on some other topic, some other dias. So like many people, like the people from biotechnology should be in entrepreneurship, so that we know the consequences, the cons, pros, everything from our field, so that we can lead our field, we know what we need. So we can tell that I faced all these things, so I can bring change here. So we need for our younger or for our juniors. So this is also a good choice. So currently, what is the fields in biotech that shows promising and good scoping feature? Like when I was wanted to join BTech genetic engineering, nobody knows what is genetic engineering, and everyone they said was like, there is no scope in this field. Whatever decision you are taking is completely upon you. But suddenly COVID happened, and now everyone knows biotech, and everyone biotech is also racing equally against as a IT company. So we can't tell what is the scope um, after 5 years. Ultimately, we are working on something like research or health field. There will be always some space or some importance given to the research. And after 5 years, I guess and I hope biotech will also be leading equally to the IT companies in India, and will get so much of opportunities. And don't think on scope, all those big words, because when we are doing something, we always aim for getting money, and will there be a scope, will I get a job? Don't go with that. Like, do what you love. Ultimately you will get the way which is best for you. So after 5 years, there will be like so much of opportunities there. And like I promise, there will be there some good opportunities in India. Thank you. It's a better to choose MTech in biotechnology or go to MTech course with specific domain. So if you're doing MTech, it is better to go with a specific domain. Like if you're someone who likes particularly agriculture or plant, if you're choosing a specific domain, it will make you enhanced in that field. And if you're going to continue that for your higher studies like PhD or something, go for a specific domain. But if you want to do some job or join some company after MTech, do it in biotechnology, because when you're doing MTech in biotechnology, you will get opportunities to learn everything again. You are not specified to one single domain here. So you'll get so much opportunities when you are going for job. And when you choose it that I'll be working on this cancer biology only, then go for a specified domain, so that you will be excelled in that particular course. So you'll be given importance there. So based on your next step, choose biotech or a specific domain according to your next, what is your next aim in your life. Fellowship JR in the immunopathology work environment perspectives in detail. So I completely don't know about this fellowship details. I'll look into it and I'll let you know. Ma'am, what kind of attitude or mindset do you suggest I develop for having a career in R&D? Like, try, as I said, uh, don't think that this is the important research. Discovering the drug is important. No, accept everything. It's like whatever work you are given, accept it and do it. Don't expect anything from it. Like if you are doing some PCR or if you are doing some experiment or even a 6 months project, you are doing something out of it. Don't expect. Like science is all you are doing is you finding something unknown. Like you are doing some research to find what is not known to others. So you are already a detective there. So accept everything, and whatever you are getting results, whatever you are facing, everything, just okay, you accept it, learn from it and move on. And like if you're asking about working environment, don't get into anything too serious. Like whatever they are doing, whatever other people are into, it's their work, and that's none of our things to think about it. So make yourself the main character of your life. Think about you. Think about how you can pass this situation happily, or don't think everything, like don't socialize. So don't overthink everything. Just accept it and go with the flow. You'll get, there is nothing like you have to maintain this attitude or mindset. Like just enjoy the work you're doing, accept your research, accept the mistakes you're doing, because some mistake you're doing may lead to some new discovery. So even if you're doing wrongly, learn from it and try to not do that next time. So accept and move on. What kind of internship do you suggest for second year students? So I suggest like attend all the internship, like keep internship certificate from all the domain, like animal cell handling, cancer assays, plant thing, and flow cytometry, microscopic imaging. So whatever I'm telling, all these are like keywords from my PhD applications. So attend internships from all the domains. Learn every techniques, because in internship you will get only one time opportunities to keep PCR. You'll get one time opportunity to see the microscope. So we are not going to excel in the internship, but at least you know what is the principle, what is the basic, how it is working, why it is working. So at least you'll have the basic idea. So attend all, like in the curriculum, in the BTech student, there will be only one internship that is to be compulsorily you have to attend. But don't do it for academic purpose. Try attending as much as internship as possible. Like when you are choosing the next step, it will be very easy and your CV will look perfect. I'm interested in both genetic engineering and forensic science. Which one do you think has better scope? So if you are interested in genetic engineering or forensic, you will have some idea like I have to do this kind of research. So if you are like interested in forensic and science, that means you have to be in some detective field or some other DNA fingerprinting, some other. So write down why you are interested in genetic engineering, why you are interested in forensic science. So when you are asking questions for yourself, why I like this, why I want to do this, you'll somehow your heart will tell do genetic engineering, because you'll always favor one step ahead, which is your heart and which you are truly interested in. So in that way, you can find out in which domain you're working. First, you try to speak with yourself and understand why you want to do genetic engineering and why you want to do forensic science. So when you keep asking this question, you will get idea like why I am interested and which one you are most interested in. Like you guys were very interactive. I didn't expect this much of interaction and questions. Like it was very happy that like you're asking the questions out, your doubts, everything. Will it be a better option to get into a job right after any BTech degree? Yeah, like if you want to do job, there is no difference that you are doing job after BTech or MTech. Even after MTech, you are going for some job, you will be given same salary, same importance as a BTech student. So they won't see that BTech will give 25,000, MTech will give 35,000, and there will be no difference in your salary package. There will be no difference in your like role you are working on. So if you are willing to do job, please do after BTech. Don't take another risk. Like even after MTech, you will be given the same importance as BTech. So if job is your main thing, you can join it after BTech itself.
Good evening, ma'am. My name is Santosh.
Yeah,
Thank you for the very informative session, ma'am.
thank you.
So, especially, I have completed my BTech and looking forward to pursue my masters. So, it will be a great guidance for me, ma'am. And I completely agree with the point you said that the opportunity to learn after your studies, especially job, is really true, ma'am. Because I'm working as a research assistant in between the gap between the masters and the BTech time. So, how can we make the learning process more effective, ma'am? In this because as you said, they're mainly focusing on their work, what is already going on in the laboratory. Yeah.
Okay. So now you decided to do Masters MTech or Masters or abroad or some other India. So
Yes.
you will be having some idea what field you have to do.
Yes, ma'am.
So what you're choosing?
I have chosen in the regenerative medicine, ma'am.
Regenerative medicine.
Yeah, yes, ma'am.
Okay. Now we are working in as some research assistant.
Yes, ma'am.
Okay. So go learn some basic or advanced, like start learning, reading the papers, something related to regenerative medicine. So if you are aiming abroad, see the labs who are working on regenerative medicine. So there will be like the best thing in abroad is every professor will have like separate website for their lab.
Yes.
Like every professors will have their separate website. So first take the list of the professor who are working on regenerative medicine, even in India or even abroad. Check out
Okay.
their research papers, recent research papers. See what are all the techniques they are using.
Okay,
Like
ma'am.
if in India you want to work, see what are all the techniques they are using, and try to see. Now, if you are working in some VIT or something, look into that professor, that whether he has any connection with VIT, any correspondence or, author or any publications from VIT, so that you can have collaborative works.
Okay.
So it will be very useful for you, because now if your professor and the professor you have chosen has some research paper, so that you can ask your professor and go start working there as a research assistant. So you can work 6 months as a research assistant and you can continue as a PhD student. In that cases, that professor will accept you soon, because you will be joining him as a PhD student.
Yes.
So he will be very interested in taking you. So if you want to go abroad, take the professor's team profile or what is the publications they have. So start learning the techniques. So if you're working in some department, try to speak with your sir and tell that you want to learn this.
Okay, ma'am.
So if a nearby lab someone is doing flow cytometry, have good connection with the PhD scholars, postdocs, everybody. So make like friendly way connection. So if your friend who is working in the nearby lab is doing some work on flow cytometry, go and take part of it. See how he is working. Learn from him. So you should have a collaborative nature. Like you have to be like try to know what is happening in other labs also. If they are using some new techniques, try to learn that technique, because when you are in lab, when you are in college, try to use all the opportunities, try to use all the equipments, try to use all the things. Like don't miss that opportunities, because no staff will say that will not teach you, you don't come to lab. No, when you're good with some PhD scholars, they will help you. Okay, because they also need someone help, right? You'll be
Yes, ma'am.
doing it because ultimately you are benefiting by learning what it is.
Yeah. Yes, ma'am.
So try making connection and collaboratives with your nearby labs to learn the techniques.
Sure.
Or see the research papers, like based on the field you have to do your masters.
Okay. Thank you, ma'am. Thank you
Thank
for
you.
valuable information. Thank you.
Thank you.
Thank you, ma'am. Thank you
Thank
for
you.
your valuable time here and clarifying all the doubts. And thank you for providing valuable insights into the real world applications and academic pursuits and industry careers.
Thank you.
Um, Thank you for your time here. If you have any doubt, you can uh, follow her and ask any doubts in LinkedIn. We'll share through mail. Now I call upon Savita Madhusudanan to invite our another guest. Thank you, ma'am. Thank you for
Thank you so much.
A very good evening to one and all present here. It is a pleasure. It is a complete pleasure to have you all here. We have a huge attendance today. So on this DNA Day, it is my honor to introduce Dr. Kriti Kaushik, a distinguished expert in the field of gene therapy, hailing from AIMS. With a relentless dedication to revolutionizing healthcare, Dr. Kaushik has strategically charted her career path to confront life-threatening diseases head-on. For over 8 years post PhD, Dr. Kaushik has immersed herself in the intricate realms of gene therapy, specializing in cancer immunotherapy, RNA DNA based gene editing, and lentiviral based gene therapy for diseases like Gaucher's. Her journey began with a solid foundation in genomics during her doctoral studies, a path that eventually led her to collaborate with various research groups and startups, unraveling the genetic complexities underlying different cancers through cutting edge sequencing techniques. Dr. Kaushik's contributions to the field are not only evident in her prolific research output, which includes 10 publications and 2 communication pieces, but also in her successful acquisition of prestigious grants such as the DHR ICMR Young Scientist Award and DBT Research Associate Award. Her academic prowess is further underscored by her qualifications having secured DSC Inspire JRF and CSIR Net JRF SIRF accolades during her PhD research. Today, Dr. Kriti Kaushik stands at the forefront of gene customization, pioneering innovative strategies that hold immense promise for the healthcare industry. Her expertise and unwavering commitment to advancing gene therapy make her a beacon of hope in the quest for combat life-threatening diseases. It is with great anticipation that we welcome Dr. Kaushik to share her insights on gene customization, a budding strategy in the healthcare industry. The dice is all yours now.
Thank you. Thank
Thank
you,
you.
Savita. And thanks, Scioverleaf team, for having me here. And I was just listening to you guys from past half an hour, and it was so exciting that you guys have so many enthusiastic questions. It's really nice, when you interact with such enthusiastic minds, and you happen to see a one-on-one connection. So really great. I must congratulate you guys for all this. And I have noted down a few questions that you guys were looking for. So the first question that was asked, somebody asked about fellowships in immunopathology. After, is that a JR fellowship that you are asking for, or a postdoctoral fellowship? May I know that? I can't hear her. Okay, so be it any fellowship. First of all, to get placed for the junior research fellowship. So be it any fellowship at any level, okay, be it at the PhD level or at the postdoctoral level, they are not subject specific, first of all, okay. You get fellowships or at the PhD level based solely on your net and your fellowships entrance examinations that you pass, and you get your postdoctoral fellowships solely on the basis of the projects that you write, which have to be translational and making some some application, so that because you have a lot of high competition, you compete with a lot of people around you, so you need to design projects which have a good outcome. So, as I said, you don't have anything in particular for a subject, okay? So you just have to make the projects. And this is, I'm talking about at the postdoctoral level. At the PhD level, you just have to give some entrance examinations, and which includes basically all the subjects, okay? All the life sciences subjects. So this one is that. I hope it satisfies your question. You can write in the chat box. Maybe we are not able to hear you. The second question that prompted me was somebody asked that should I go for MSc in bioinformatics? So going into bioinformatics at the MSc level will help you if you are looking for a job right after masters and you're not looking for any research role. One. Second, if you're kind of looking for a long career into research, like after masters you want to go for PhD and all that, I would suggest you that MSc in bioinformatics would limit your options. So go for a basic research or something like biochemistry, biotechnology, something that has a broader category. So that way you are eligible to apply for PhD positions or other research positions, you have a broad spectrum to apply for. One more thing is that the tools that you get to, because I have been through a little bit of course curriculums of MSc Bioinformatics taking place in different universities in India. So the courses that, you guys have, the course curriculum, those tools you can even learn after doing your masters in case your field requires that. So just based on that, you don't have to do your MSc Bioinformatics, okay, because that really limits your options. So go for a broad spectrum course at the master's, at the master's level. Okay. So yeah, and somebody was asking about primer designing. So in case I have time after my session, I will give you a live session on how to design primers. Okay. Is that fine with you all?
Sure, ma'am. Sure, ma'am. We'd be glad to have you.
Okay, so I'm just sharing my screen, and please let me know in case everything is visible. Okay, are you able to see my screen?
Yes, ma'am. We are.
Yeah, my slides are there.
Yes, ma'am. We are able to see.
All right. So today I'm going to talk about my very favorite subject, that is gene customization, which is also coming out to be a budding strategy in healthcare industry. And I have tried to gain some subject matter expertise on the same from the past 8 years, and right now I'm trying my hands in academia and trying to switch my gears to industry. Let's see how it works. So first of all, let me let this be an equal interactive session. So what is gene customization? Okay, so we all know what by this, as the term is suggesting, this is like the defective gene, is tailored. And how it is done, sometimes the gene gets defective by a point mutation, so either by correcting that point mutation, or you replace the entire deceased gene with a healthy gene, or you silence the gene completely. Okay, sometimes you don't want that gene to express at all. In case of cancers like oncogenes, you don't want those genes to express at all, so you silence that gene. So in either of these cases, something that you're doing by tailoring a gene is known as gene customization, often known as gene editing or gene engineering. All these three terms are same. And precision medicine approach, it is a precision medicine approach. And why precision medicine? Because we all are humans, but we all differ from one another. We all differ from, I mean, we are not identical. We don't look identical. We have different kind of, we are susceptible to different types of diseases. And why this happens is because we, despite having the same 3.3 billion base pairs of DNA, we differ from each other because, there are single nucleotide variations. Sometimes there are different types of polymorphisms that we all have, which makes us different. That is why we are susceptible to different diseases. We look different, and so we have different heights. We have different features, right? So that is why we all have a specific DNA, although we all belong to the same human mankind. So precision medicine approach is to change the specific DNA sequence in an individual and customize the genetic makeup. So it is simply a cut and paste mechanism of engineering the gene. Now, why do we need to customize the gene? Like, what is the need after all? So you can see a bunch of people here on the screen. So they all are facing the same disease, okay? Let us imagine they are all having the same disease, but they are grouped into 4 different categories. Why? Because although they are having the same disease, but because of their different genetic makeup, the causative gene in the 4 groups are different. And that is what is making these 4 groups different from one another. Okay. So any disease, you say, like they can be caused by multiple genes. Okay. So we need to customize. So when precision medicine is when we study the genome genetic makeup of an individual, and based on the causative gene, you treat that individual. Okay, that is precision medicine. And it is not like somebody has come over, like somebody is, so it's simple, like okay, everyone understands how cancer is caused. Okay, now if you go to any hospital for the first line treatment, okay, the first line of treatment, that is general, without understanding the genetic makeup of the individual, it is like either surgical resection of the cancer, and if it is not possible, chemotherapy is given. It is very general. Okay, so these 2 first line of treatment doesn't involve the studying the genetic makeup of the individual. Okay, it is only at the later step, at least in India, when genetic makeup is studied, and that too in India it is studied at a later stage because it has a lot of constraints of the cost and it's not cost effective. So but these types of treatments are becoming very well known in the Western world. Okay, so once you understand the genetic makeup, you are able to tailor the right gene and able to treat the disease. Okay, so this definitely helps to cure the disease by correcting that mutation, and obviously it stops inheritance of that faulty gene into the other lineages. Also, it is done to sometimes, when you are into a lab and when you are into research, you want to understand the role of a specific gene in a disease. You don't know, we just know, okay, probably this gene is going high in this disease, okay, but we don't know what exactly it is doing. So what do? You just try to silence that gene, okay, or you try to overexpress that gene. Basically, you try to modulate the expression of that gene, okay, and you do this all by customizing the genes. So you want to understand the role. So when the gene is silenced by knockout technology, you observe the phenotypic changes, and then you go to the genotype, and then you see what changes it has made in the pathways and cell signaling and whatever. Okay, so it also helps to understand the role of a specific gene in a disease. Also, at the same time, when you want to model a disease, so for example, I already know that this particular gene is causing this particular disease. So I want to model that gene and then apply all my potential therapeutic strategies that I'm trying to apply on that. So I want to model a disease. So mutative genes are incorporated into a wild type model organisms or cells, and then you try to customize that gene and see whether your therapeutic strategy is working or not. So I will be talking about this particular point in one of my slides later on, where I'm trying to do a particular work here in my lab. So before that, let us understand how is gene editing performed. Okay, so there are 4 major approaches right now, and I'm sure you guys must be aware of few of them, some very popular terms going on these days. So the first one are the nucleases. So the nucleases are the category of gene editors which create double-stranded breaks, and the techniques that fall into this category are zinc finger nucleases, talons, and CRISPRs. CRISPRs being the latest one and quite in fashion these days. Okay. The second category belongs to the base editors, where they try to do single nucleotide substitutions using a dead Cas. So Cas is a nuclease. Okay. So what we do is it's known as a dead Cas because it is not able to create a double-stranded break. All it is able to do is that it is able to recognize the target well. Okay, so we use a dead Cas, we use a guide oligo which will take us to the target, and we use a deaminase with this. Mainly these deaminases are cytidine or adenine deaminases. So they change the mutated base C to U or A to I mutation. Okay, so that is how you're able to create the single nucleotide substitutions. Then you have prime editors. Okay, prime editors are the Cas9 nicases. So they are not able to create a double-stranded break, but they create a single-stranded break. And they carry a guide RNA, okay, which has the correct copy of the gene. So what happens as soon as it is able to create a single-stranded cut in the DNA, the reverse transcription puts the corrected RNA. So the reverse transcription actually transcribes the RNA into cDNA and incorporates the edited copy of that cDNA at that particular place. Okay. And then you have the fourth category, which is very much a rare category right now, which is a paste mechanism, which is a drag and drop editing. So this is mainly for large insertions without creating any double-stranded breaks, and they use a serine integrase enzyme, and they can integrate up to 36 kbs of DNA without creating a double-stranded break. So the last two techniques, basically the prime editors and the paste technology, are mainly in very early stages right now. So they are not very well developed. So people are working out because they have their own challenges. But the main ones are the nucleases and the base editors, which are playing a very important role these days in healthcare industry. So I will shortly explain you the kind of work that I do, and I'll show you some of my preliminary results. I'm not giving you a detailed description, just an overview. So first, I'm basically doing both DNA editing and RNA editing. So we use CRISPR-Cas technology and a lentiviral system, which is my delivery mechanism. Okay. So the first step that when you do a DNA editing by CRISPR-Cas is how, first of all, you have to design guide oligos. Okay. These are very small oligos of 20 bases, and they are designed so that, you know, they are designed at the target site of the gene. Okay. So as you can see, I have put up a gene cartoon over here, and the orange bars are my exons, which are also the functional region of my gene. Okay, because these exons are going to get transcribed into RNA, and then ultimately part of it is going to get translated into proteins. Okay, so when I'm designing the guide, either I have to design it at the AUG codon, okay, so that I just stop its translation, or faulty protein is produced, okay, so that my entire protein is not produced at all, or I design it anywhere in my functional region. Okay, and how the guides are designed, they are not designed randomly. So the kind of Cas that you're using, in this case I'm using a Cas9. Okay, so they recognize some PAM sequence, okay, protospacer adjacent motifs. So for Cas9 it is NGG sequence. So these are the triplet sequence where N is any nucleotide followed by 2 Gs, guanines. So wherever in my functional region I'm going to find that NGG region, I have to design a guide 20 bases upstream to that, okay, which is of 20 bases long. Okay, so it is designed right upstream to that TAM sequence in the functional region, and you have to make sure that your guides is targeting specifically to your own gene and not to any other gene. Now, once you have designed the guides, so what I do is I clone them in lentiviral vectors. And the reason cloning them in lentiviral vectors is that the lentiviral vectors, when they go inside the cell, they make an integration. Okay, so that's a permanent integration into the cell. Okay, so my guide is now going to be constitutively expressed in my cells because it has integrated into my genome, and it has its own mechanism of transcribing and translating. That's it. So that is why lentiviral vectors are preferred. So guide oligos are cloned into these lentiviral vectors, and we basically produce virus, okay, by transfecting this clone into HEK293T cells, because they are the cells which produce virus. And then we collect this virus, okay, and the next step is the target cells that we are targeting. So basically I work on leukemia. So I have some leukemia cells which are already Cas9 expression, and that also we have created by cloning the Cas9 gene in a vector and then transducing it into those cells so that I have a constitutive Cas9 expression. Okay, so like that I have Cas9 expressing leukemia cells, and I have virus from my guide oligos. So I transduce it into these cells, and what I get is, so I have a fluorescent marker for the vector in which I have put up my guide oligo, one fluorescent marker, let's say RFP, and my Cas9 cells are already having a GFP expression, green fluorescent protein. So ideally, when I will see my final cells under a microscope, I will see both GFP and RFP. Okay, and the dual positive cells are the ones which have both my guide oligos and Cas9. So those are the cells where my gene editing is definitely going to take place. Okay, and once I'm done with this process, now I have to reassure that my experiment has taken place correctly. So I check it with a flow cytometry, and I can use this machine only because I have used some fluorescent tag markers, which is GFP and RFP. So I need to sort them out by my flow cytometry and facts, that only dual positive cells. I confirm it by Sanger sequencing as well. Okay. And then I confirm it by a PCR. Okay. So after this, these are my results. Okay. So I'm trying to knock out my gene X in my Cas9 expressing cells. Okay. And if I can, I'm trying to, yeah. So if you can see, these tiny dots are my cells. Okay. So this is a 10x expression, and very small cells, these are my leukemia cells. And they basically, because they are the blood suspension cells, so, the aggregates, they form aggregates like this. Okay. If you can see this, I can't zoom it more than this. So what I've done is I have taken my cells. So mock is something where I have not added anything. So you need to have a blank in your experiment, a mock in your experiment, because you need to compare it with your test. Okay. So I have not added anything in these cells. These are simple, my Cas9 expressing cells. Then I have my NT, which is my control. That means there is a guide oligo that has gone inside these cells, but it is not targeting my gene X or anywhere in my genome. So it is a scrambled oligo, which I have put inside, but it will not target any part of my genome. Okay. So this is my control. Okay. So one is my mock that acts like a blank. Then is my control, because I have introduced a guide oligo, but it is not going to target any of the gene. And then I have my two guides, okay, which are specifically designed for my gene X. Okay. So now that you can see that my cells are expressing both GFP and RFP. So I need to sort them out. Okay. Because there will be some cells which will be only Cas9 expressing. Okay. And probably my guide has not made through it. So I need to filter out those cells. I need to remove those cells, and I need to save only those cells which are dual positive. So what I did a flow cytometry sorted experiment, and this is the kind of graph that I got when I just ran my cells into it. And so you can see this second quarter. This second quarter is my dual positive cells that I have sorted out. Okay, these are GFP, RFP positive cells. So only these cells have been sorted out. Okay, rest all have been chucked out. Okay, and once they have chucked out, what I did is I tried to check the protein expression of my gene after knockout. Okay, and this is what I got. So I used a internal control gene, which is a GAPDH. Okay, and it is a housekeeping gene. So to make sure that my experiment is fine and there are no changes in the protein levels that I have loaded onto this, so I have used this gene. As you can see, the GAPDH in all the 4 samples is same, which means I have not by any way manipulated the amount of protein that I'm trying to load. Then I'm trying to see the expression of my gene X, which has been knocked out. So you can see that there is a basal level expression in the mock. And then again, there is a basal level expression in the NT. But you can see a very faint expression in the 2 guides. Okay. So there is a significant knockout that and this is before my sorting. So I have not put up the result after sorting over here. So definitely I won't be able to see any protein expression here after my sorting experiment. Okay. So so this is how it works. So this is, and after that I'm going to go for downstream analysis for my gene X, whatever it must be used for, or whatever role it would have been. Related assays would be performed. Okay. So I have successfully knocked out my gene here. Okay, great. All right. So this is the preclinical side that I've tried to show you through my experiments. But right now, the DNA editors have moved far off in the clinical trials. So there's a good news that we have one CRISPR-Cas gene editor, which is designed against the sickle cell disease by Vertex Pharma, has been FDA approved just 4 months back, which the first drug, which is Casgevy. And there are a number of drugs which are in pipeline from CRISPR Therapeutics, Caribou Biosciences, Vertex Pharma, Editas Medicine, and Beam Therapeutics. Okay, and one of them is in a very advanced stage by Vertex Pharma. Okay, it is being used for beta thalassemia. All right, so you can see that this field is really progressing very fast, and we hope that we get good editors out of it, and it's a useful asset to the mankind. But with this technique, there are set advantages and disadvantages. So the pros are that definitely it helps to treat the diseases, and obviously it helps to extend the lifespan because you're trying to customize your gene. But at the negative side, there are a lot of ethical concerns, because you're trying to manipulate the genome, and you're also trying to reduce the diversity, because the very first thing that I said was we all are different from each other because we have the same genes. I mean, but those genes differ from one another through those point mutations and little bit of differences. If we try to correct those mutations, then definitely the diversity between us will reduce. Okay, so we are trying to go against the nature somewhere. Okay, so it's good as far as we are able to correct the disease, but otherwise we should not be reducing the diversity. It should stay the same. Then definitely there are safety issues, because so most of the delivery systems that are utilized for CRISPR-Cas are the viruses, the lentiviruses and adenoviruses. But lentiviruses, as I told you, integrate into the genome, and then they start expressing that gene. So there is a permanent change into the genome that you're doing. Okay, and that permanent change could be anywhere in the genome. So there are chances that it might convert, that integration might convert a tumor suppressor gene to an oncogene. Okay, so due to those disruptions, that could lead to other severe health defects. Okay, and definitely there is a high cost issue. So right now the treatments that are available in the market, they are very, very costly. Okay, so high cost is definitely a concern, but let's see how it moves in the future. So the second strategy that I use in my lab is RNA editing, which is again by CRISPR, but a different type of Cas this time, which is Cas13b, because we are targeting the RNA. Cas9 targets DNA, Cas13 targets RNA, and we use base editors. So here, so the DNA editing part I'm trying to do on my cancer model. Okay, and this RNA editor is something I'm trying to do on a rare disease, which is Gaucher disease. And this is an autosomal recessive disorder, which is caused by a biallelic mutation of the GBA gene. And the current therapies are either expensive or very difficult to perform. So the objective we have is that we need to establish an ex vivo monocyte to macrophage model system from GD patients, because this Gaucher is basically affecting macrophages mainly. So we are trying to establish an ex vivo model system from the GD patients directly, and then to correct the mutation by overexpressing the wild type gene and RNA editing the mutated gene using CRISPR-Cas13b system. So as you can see in my methodology, the first part that we are trying to do by creating an ex vivo model system is, we have a GD patient and we have the human control. We isolate PBMCs out of them, and then the monocytes, the peripheral blood monocytes. Okay, so we grow those monocytes, and then we differentiate those monocytes into macrophages. So we have got the blood out of the humans and trying to culture them outside. So that is why it is ex vivo. Okay, and using a differentiated marker, which is GM-CSF, we convert it to macrophages, and this entire process is 10 days long. And then we verify whether our macrophages have been differentiated well or not using a flow cytometry experiment. And the second thing is that we are trying to overexpress the wild type gene and correct the mutated gene using a CRISPR-Cas13b system. So what we do, we have taken again the genes from both patients and humans, the GBA gene, the causative gene, and we try to clone it in another lentiviral vector, which has a GFP marker. It also has a RFP marker, but please remember, this vector is different from the vector that I have used in my previous DNA editing system. In this case, if I clone my gene into this vector, it will show GFP, but after cloning, that RFP will go away. Okay, so it is different. So when I have cloned my wild type gene and mutated gene, I verified it by Sanger sequencing. And after that, I have put my clones and developed virus out of HEK293T cells and again transduced it into the cells, HEK cells only, because I'm trying to create an ex vivo model. I'm trying to model a disease apart from macrophages. I need to have a proof of experiment that if it is working in HEK cells, definitely the same experiment should work in macrophages. But macrophages are difficult to transduce. So before that, I need to put a proof of concept of my strategy. So that's why I'm using cells which are easier to transduce, which are HEK cells. So I produce a virus and then I tried to correct the gene using CRISPR-Cas13b. Okay, and I have some preliminary results about it. So first of all, when we develop monocyte to macrophage differentiation from the patient cells and the human control, so you can see here the morphology of the cells. You know, this is like from day 1 to day 10. And if you can see from day 1 to day 10, the cells have grown. Okay, they have become larger. And if you can see morphological difference, you will see between the healthy control and the patients. Okay, so you can see some elongated cells and crumbled cells in the patient samples in comparison to humans. So that is one morphological appearance of Gaucher cells. Okay, and then we took these cells at day 10 and tried to do a flow cytometry experiment out of it. And so basically by flow cytometry, so there are some markers which are expressed by macrophages after differentiation, which we can clearly see here. They are CD11b positive, CD14 plus, and CD16 plus plus. That means they highly express CD16. Oh, sorry, highly express CD14. Okay, so we confirm it by flow cytometry. And then we come to the second part where we try to model this disease in HEK cells. And as I told you, this vector, once it is cloned, the RFP will go away. So that is why when we clone the wild type gene and the mutant gene, and we put it into the cell, we could see the GFP expression, but there is no RFP expression. And when we use the vehicle alone, because it has no gene, it is just the vehicle vector alone, it is showing a RFP expression in the cells. Okay, so after correction, what we did is so just a minute. After the correction, we tried to again. Yeah. So we tried to do. So we confirmed this by using a Sanger sequencing. So there was a mutation. Okay, and then we checked for the protein expression. So there was a high protein expression in the wild type gene and lesser in the mutant gene. And also we checked a GB activity assay and we found that the activity in the mutant GBA cells were 6 times lower in comparison to the wild type. So again, this is my preliminary results, and I have some more results over this, but we have performed those experiments only once. So we are trying to replicate it in more biological replicates. So I've not put them over here right now. So RNA editors in clinical trials, there are a lot of them, but as you can see in comparison to DNA editors, so for the first drug or the RNA editor which has got the FDA approval for entering into the clinical trials is this Wave Life Sciences product WVE006, and it got its approval just few months back, some 7-8 months back in 2023. It is in phase 1 trial, clinical trial right now, and it is against this AATD disease. There are 2 more editors, RNA editors, which will be coming up in clinical trials later this year or in early 2025. But rest of them are all in preclinical stages, and 2 of them are coming up from Edigene and ShapeX Therapeutics, but they are not disclosed currently. So, but you can see that there are very few RNA editors, but if they come over, I'm sure they're going to take up the healthcare industry. So the advantages are that this technique is highly specific, definitely, because it's reversible, it's versatile, because it's not making any change in the DNA. It is only at the RNA level. You can tailor the RNA without creating any genetic change. But the cons are that it can have off-target effects because there are delivery challenges. There are no proper delivery mechanisms, and that is why the how to target at the right place, that's the reason why it can have off-target effects. And obviously it has limited applications because it is targeting only RNA. Okay, so some RNA have a very short half-life, and some of them have longer ones. So you cannot target every RNA, and then also you can target only those where you are basically having point mutations. So in this case, in our case, we are using adenine deaminase enzyme, okay? And basically we are trying to correct the gene mutation from C to U, okay? But that part I have not included over here because that's right now in more replicates. So the future of gene editing is that we need to design definitely efficient delivery systems so that the off-targets effects are minimized, and disorders like Huntington's disease and Alzheimer's can be corrected. Preclinical studies are going on to use CRISPRs against the infectious diseases, definitely, like against bacteria and virus. And there are some studies where they're trying to use it against HIV. So if you can, modulate the CCR5 receptor of HIV, you will be able to stop the propagation of virus. Then DNA polymerase editors are coming over, which can overcome prime editing by introducing large insertions. Epigenome editors are coming. And definitely AI methods are being used to personalize the gene editing approaches, creating more therapeutic applications. So the power of AI, as you guys must be knowing about, so it's being harnessed for the engineering of novel, more efficient, and specific nucleases through novel computational protein design methods. So this is it, my very short presentation, and thank you very much. I'm open to questions. Yeah, just a minute. Yes. I can have questions. Yeah, please go ahead.
Ma'am, I think she has actually commented the question. Vectors are mainly for the expression range of disease, combat at first encounter. How do we confirm it in the redesigned gene of the alive subjects?
You mean to say humans?
I'm not sure, ma'am.
Vectors are mainly for the expression range of diseases, combat at first encounter. Didn't get your question actually. How do we confirm it in the redesigned gene of the alive subjects? Can you simplify your question? I think so. What I got, any live subjects tested in the so that's going to happen in the clinical trials. Now, so these are just the preclinical studies which happen only on cells or model organisms, and then they are approved. They have to get an approval to test it out in alive subjects, like, you know, for humans. So for that, they need to go to the clinical trials. So that's why I showed that table, that there are very few subjects in clinical trials right now. Does that answer my question? You can you can pursue your MTech in genetic engineering. Can CRISPR-Cas be used to treat gliomas? Yes, of course. It can be used for both solid tumors and for hematomalignancies. For all kind of diseases, it can be used. Yeah.
Do we have any other questions?
I hope you guys have understood it, because I've tried to make it simple, okay? And I hope you guys get it. I know you.
Just interested in primer designing.
All right. How is CRISPR used in researches like stem cell therapy? So basically, they use CRISPRs to engineer the stem cells and to differentiate them into the cells of a specific lineage. Okay. If there are no more specific questions on this one, so I can show you guys some primer designing. Is that okay with you guys, or you have any other questions?
Yes, ma'am. That is totally.
Okay. I'll just open my few browsers and then show you. So most of you are in bachelor's. Do we have masters also? Master students over here? Most of you are bachelor students.
Actually, yes, ma'am. A lot of the participants here are my juniors too, from my college.
Oh,
Yeah.
so which year are you guys in? So right now.
I am in third year, and all those participants, they are from first year and second year.
All right. Okay. So I'll show you. Okay, I'll show you directly then. I'll present my screen. So before we go through that, can you guys tell me that why do we actually design primers? Why do you want to learn this? Any one of you can raise your hand, unmute yourself, and go ahead. You want to learn something without any purpose? You must be knowing now. Why are you going to use this? Or is that a fashion technology? Yeah, PCR. Right. To run PCR. Yes, that's right. So, okay, I'll show you. So give me a gene that you want to design for them. Give me a gene. Any gene that is coming to your mind? P53 HER2. Okay. All right. So you guys can see my Google screen here, binge screen.
Yes, ma'am.
Okay. So the first thing that you're going to do is there are these genome browsers, okay, where you can get hell lot of information about any genome and any gene. So this is like very comprehensive kind of a thing. Ensemble genome browser, and it has the latest version of 1.11. So you click on this. Okay. Okay. One more thing that you have to keep in mind is that if you do it, since we are in India, if you do it in the later part of the day, you might face some, crunches uh, during this time of the day, because this is European, okay? And most of these genome browsers are controlled, like it's like going on and on for like endlessly. So people right now in the Western world are heavily using this, okay? So they, because this is using the European mirrors. So that's why we face a little bit of connectivity problem during this time. You can go for UCSC also. So this is another browser that we use, okay? So I have specifically typed UCSC, but you can go for gene search also, UCSC genome browser. I'm just trying if I got through ensemble. Good. So here you need to click on human. Okay, select the human genome or whichever genome you are working on. Okay, and you click on this. Actually, I would have explained it nicely, because in ensemble gives you a full, detailed this thing view of your exons and introns, and it gives you a good this thing of where you should design your search. Oh my god, I'm having a tough time through my internet. If you guys give me 2 minutes, I'll change my internet connection. If you guys don't mind, and you guys are really enthusiastic to learn about, I'll just change my internet connection and get back to you. Okay.
Yes, ma'am.
Okay, can you guys hear me? All right.
Yes, ma'am.
Okay, so yes. So I clicked on that human genome, and this kind of a window will appear on ensemble genome. So here you can click your gene of interest in this particular box where search is written. You just type your gene, let's say HER2. Okay, and go. Okay, so a lot of this thing will come over. You need to select the HER2 gene. Okay, so you need the transcript. So a lot of options are coming. You need to check the HER2 gene. Okay, so there are gene categories here, 3 gene categories. You need to click on that on the left side. Okay, and the second category tells me HER2 gene, not by this name, but in the other gene names, HER2 is coming. So I click on the second option here, which is ERBB2. And this kind of a window will come where it will show me everything about my gene. Okay, so you can see the gene synonyms, HER2 is coming. Show transcript table. This kind of a transcript will come. So a lot of transcripts are there, but there is only one main transcript that is getting translated into my protein, and I see that by my RefSeq match, which is my NMID. Any transcript which has a NMID, that is the one which is getting translated into protein and is the causative gene. So I click on this transcript, okay? And when I click on this transcript, the window that will show me will be the summary of this transcript, how many exons and introns it has. So on the left-hand panel, there is under the sequence category, there is exons, okay? So you click on exons and you click down, okay? So you can see there are 27 exons, okay? Each exon has a ID. And when you design primers, okay, it's always ideal to design primers at the splice sites, okay? And in case you want to amplify the full gene, you need to design the primers end to end, okay? So that you cover the entire gene, okay? End to end. Otherwise, if you're doing it for real-time PCR, which is quantitative PCR, I always advise you to do that for splice site junctions so that only your cDNA is amplified. Okay. So right now I'll try to design it anywhere in the gene. Okay. So a good primer is of 20 bases and it should match only to your gene of interest and not to anywhere else. Okay. So I will take up 20 bases anywhere. Okay, here. So randomly, I take some few bases, okay? And I should make sure that this is at least 20. So I copy this and I go to my previous, this thing, where I was using UCSC BLAT. I will go to UCSC BLAT. Now, what does UCSC BLAT do? UCSC BLAT will tell me that my gene, this particular primer is only binding to my gene or to any other place. So when I submit it, I get only one hit. So it tells me that my query, that means the sequence that I submitted is of 21 bases and it's only single. It is not mapping to any other part of the genome. It is only mapping to chromosome 17 on the positive strand from this coordinate to this coordinate. You can verify this, that the HER2 gene is on chromosome 17 and the sequence that I have picked up is this, okay? So there it's fine. So this can go directly as your forward primer, okay, which binds to one strand of DNA. Now the second thing is that you need to design the reverse primer, okay. So, and all right, one more thing is there is oligocalculator, okay. So when you put your sequence into this, it will also give you the percentage of your GC content in your primer. The GC content in your primer should be always between 40 to 60. Okay. So you calculate it. It is showing you a high GC content, which is 67%. I don't think so. You should go ahead with this. What you can do is again go back where you selected it. Okay. And yeah, I selected this here. So I need to definitely, look for a position which is less in GC content. Okay. So I will maybe I will try to select some few bases from here, which I can see I have few T stretches, so I shouldn't be able to. So again, I do the same exercise. Okay, I paste it here. I submit it. I'm getting a unique match. I go to my oligo calculator. I paste it here. I calculate. I see a 48% GC content, which is good to go. I can go ahead with this. Okay, because it is falling between the range of 40 to 60%. Next, I again go here. I choose another region now downstream to it so that I can, design a reverse primer. So let's say I'm going to my third exon from here. Okay. And I have arbitrarily chosen few bases. But this is the reverse primer, so you need to do a reverse complementary for this. So before that, you need to do the same exercise. You need to check it out on the BLAT, that it is mapping to the unique location. Okay, it's mapping to the unique location. You need to check it on the oligo calculator. It has 60%, which is within the range. And then you just need to go to the reverse complement tool and paste your sequence here. Okay, do a reverse complement. This one is your reverse primer. Okay, I hope I have made it. Are you guys able to get it? You have to use 3 browsers for this. In Ensemble Genome browser, you collect the gene sequence through it. You go to UCSC BLAT to check it is mapping only once to the location. Go to the oligo calculator, look for the percentage of GC content, and then finally reverse complement it in case of a reverse primer. Okay. Hope that suffices.
Thank you so much, ma'am. That was very clear. And your whole talk was extremely, I found it amazing. And as a blog writer myself, I write articles on all these research papers, and it was really nice to look behind the curtains and look at all the analyzing properties, and it was a great impact on
Thank
me.
you. Thank you so much. Thank you for having me here. Thank you.
Well, I definitely have to mention this. You said yes to me in such a short notice, and that was something big to me. We had planned this whole event in a very short notice, and thank you so much, ma'am. It would not have been possible without you.
My pleasure. My pleasure.
Okay.
Yeah.
Thank
Great
you,
to
ma'am.
interact with so many of you, so many questions. I'm so excited that you guys are so enthusiastic and so curious to build up your nice careers ahead, and so mindful. Really, really happy because I find that in every generation I'm missing those things. I'm missing that enthusiasm. But great to interact with you all and be focused. I'm sure you will be on the right track. Get connected with the right set of people who can guide you, and no worries, be open. I have one more thing. Somebody asked that what kind of an attitude they need to have in case they want to go for R&D. You need to have patience.
Oh, yeah.
Okay, you need to have patience. And so my career in R&D is like, I started off, so I did my master's in research only from AIMS Delhi. I did my master's also from here. And that's like since 2008, I have been into research. Okay. So you can understand that how many years now, 2008 to 24, almost 16 years. Okay. And you need patience. That is one more very important thing. And you need perseverance. You need to have a positive attitude. That's it. Be positive. Okay.
Thank you so much, ma'am. We're so grateful for you, and we're completely honored to have you. And my team is probably celebrating behind. So,
I'll share my LinkedIn profile here. Somebody is asking me, so I'll definitely share it. And yeah.
so once again, Thank you, ma'am. And we'll also share all the contact information in the WhatsApp group. So
Sure.
participants need not worry.
Yeah.
So moving on, thank you again, ma'am. I could not say thanks enough.
Yeah. No problem. No problem. Yeah. All right. I'll have it then. I'll leave it from now.
Sure, ma'am.
Okay. Okay.
Yeah.
Chalo. See you. Bye. Bye. Bye.
Bye, ma'am. Thank you. Thank you, ma'am.
Yeah.
So moving on, we have one more presentation from our chief coordinator about Scioverleaf. So if you could spare 10 more minutes of your time, I want to introduce Tarangini Balasubramani, our chief coordinator at Scioverleaf, to go ahead with the dice.
Just give me 5 more minutes of your time, guys. I'll just share the presentation and let you know what we are all about. So, discovering truth is simple, and the key is to explore. We're a group of passionate students who wanted to contribute to the field of biotechnology while simultaneously expanding our knowledge. And we also wanted to provide a platform for like-minded people to do the same thing. So we started our journey on LinkedIn, posting concise blogs on research articles, but we came together and started a blogging site, which is actually Scioverleaf today. Okay, I'd like to introduce our team now. So this is Tamil and Savita, our founders. They're relentless people who have a very keen interest in molecular biology and genetic engineering. They say necessity is the mother of invention, but I'd say curiosity is the father. Tamil is driven by curiosity in the realms of genetics, and he founded Scioverleaf, a platform which he dedicated to advancing the scientific knowledge. And our other founder, Savita Madhusudanan, is all about biotechnology. Every challenge is this mystery of genes and molecules to her, which she wants to solve, and which is why she founded Scioverleaf. So moving on, our chief coordinators. Satya, she has unraveling passion in unraveling mysteries of illness, and she's an avid fan of House MD. She wishes to pave way for groundbreaking discoveries in the realm of diagnostics and treatments. And myself, Tarangini, I'm the other chief coordinator. I'm a bibliophile at heart. And whether it's literature or academics, you can always find me with up to something. I tend to focus mainly on cancer biology, but my teammates say that I'm an all-rounder. And I'd like to introduce you to the backbone of our organization, our content writers, Shazia, Vishwant, and Vaishnavi. So we also recently organized an event, Aware Her, Empowering Health and Celebrating Strength, where we had the privilege of hearing from Dr. Sudha Raghavendran. She's a teacher at Indian Culture Yoga, Lal Bahadur Shastri Center of Indian Culture, Embassy of India. She shared her insights on yoga for menstrual disorders. We also had engaging presentations by our team where they spoke about cervical cancer and osteoporosis and how these serious but conquerable challenges can be overcome. You can, so you guys can follow us on LinkedIn and Instagram, and we have a short clip for you now. Okay, so this is the link to our, this is the QR code to our YouTube channel. Our dream is to make research papers accessible and understandable to all of us. So you can subscribe to our channel, and thank you so much for attending the event today. We were so glad to have you guys here.
Thank you, Tara. That was amazing. And I did not expect all the intros. Okay, so I've uploaded the feedback link on the comments on the chat session. People who want certificates, please do go through it. Thank you, Mahiwash Khan. It was an absolute pleasure to have you all. So the feedback form will also ask you to say yes or no for the certificate. So if you need a certificate, please say yes in the feedback form. We really appreciate your feedback for further events. Thank you, Fauzian, as it was a complete pleasure to have you here. So if you fill the feedback form, you can be. And it's almost 8:30. I definitely have to say this. Thank you for saying this long. I know the event was supposed to be only till 6 to 8, but the event was very interesting. Our speakers were amazing. I could not thank them more. And thank you all for your presence in the event. It meant a lot to us.